Good afternoon, students. Today we are going to study the worms. These parasites of kingdom Animalia, they are platyhelminths and nematodes. We belong to the class helminths. Okay. Now this phylum Nematoda. In this phylum, we have all the worms which are thread-like. These worms are different from what we have studied earlier. The earlier parasites, what we have studied in Protozoa, they are quite different from this phylum, Nematoda. Now, this nematodes, they belong to the kingdom Animalia, subkingdom Metazoa, phylum Nemathelminths, and the name nematodes has been derived from the Greek word nemat, nema means thread. All these worms look like thread, long, thin, almost many are colorless. Therefore, the name has been given nematodes. The characteristics of all the nematodes are almost similar. All the nematodes, they are unsegmented. They do not have any appendages. They are bilaterally symmetrical. All nematodes, they possess head and the head is small. It has got sense organ near the head and it has got a tail. The organism is pointed at both the ends, or you can say the organism has narrow ends. One is the anterior end, other is the posterior end. Anterior end has got the sense organ or the mouth-like structure, and the posterior end has got a tail. Most nematodes are below 5 centimeters length, but as far as the size of these nematodes are concerned, they can vary from microscopic to 1 meter. All nematodes, they have got a coelom, and it is not an actual coelom, it is a pseudocoelom. This pseudocoelom is filled with a fluid, and this is going to provide an hydrostatic skeleton to this nematode. The body of the nematode is covered with a cuticle, and this cuticle is made up of a collagen which is secreted by the epidermis underlying. This collagen or this cuticle has 3 layers: cortex, matrix, and fibrillary. This cuticle is going to help this nematode to resist the pressure that is created by the fluid. That is the hydrostatic pressure that is created or exerted on the cuticle by the fluid that is resisted by the cuticle. The, because the pseudocoelom, it has got fluid. So whenever there is a fluid and whenever there is a movement of the fluid, the pressure is formed. So the pressure has to be has to be resisted. Otherwise, it will break. Therefore, the cuticle has got the character of withholding the pressure. Beneath the epidermis, there is a layer of longitudinal muscles. And this layer of longitudinal muscles, they are made up of spindle-shaped cells. And this spindle-shaped cells, they unite to form the longitudinal muscles, which are present below the epidermal layer of the cuticle. The muscles in the nematodes are not arranged in antagonistic ways, and the antagonistic role is played by the cuticle. The contraction of the longitudinal muscle on one side, it transmitted through the hydrostatic skeleton that is present, and the cuticle stretches on the opposite side. That is how the movement of the worm is possible. When the muscle relaxes, the cuticle contracts and the body returns to its resting position. Nematodes have complete digestive system which comprises of mouth, muscular pharynx, intestine, rectum, and anus. Some of the worms have teeth or cutting plates in the mouth, and their midgut is lined by a single layer of columnar epithelium or cuboidal epithelium. Now this is the picture. In this picture you can see at the anterior end there's a mouth, and this have got lips, papillae, buccal capsule with teeth or the cutting plates. All this is present in this particular area. Esophagus may be cellular or muscular. Say this one, this area. Okay. And this esophagus becomes bulbous in the posterior area. And the alimentary canal, which is a simple tube and ends in an anus. Below the anus, there is a cloaca that is present. This is the common character of all the nematodes. And one more thing is very good about this worm is that it has got a nervous system, and it has got 6 longitudinal nerve trunks. You can see here. These are the 6, 1, 2, 3, 4, 5, 6. These are the 6 longitudinal nerve trunks and circumesophageal, that is this round, circumesophageal nerve commissioners that are present. Few ganglia and sensory papillae are present on the cuticle, and that's how the nervous system is active in this particular parasite or in this worms. Another character of this class Nematoda is that the males are smaller than the females, and the males have got tail. This tail is a pointed end, pointed posterior end, which forms the tail. In males and females, both have reproductive system. So reproductive system is good, digestive system is developed, nervous system is developed. So this worm has got almost all the things that are required for its development. In males, the reproductive system consists of testis, vas deferens, seminal vesicle, ejaculatory duct which opens into the cloaca. Accessory sex organs are present, that is spicules, copulatory bursa, caudal alley, or genital papillae that may be present different species. In females, it is depending upon the species. They may have one or two tubular ovaries. Now, this tubular ovaries lead to a united uteri. If two tubulars are there, then they will unite and a single uteri is there. Okay, so this uterus or the united uteri, they will open into the exterior through an opening known as the vulva. It is also known as the genital pore, and this genital pore, it is opening on the anterior end, not on the posterior end. That is the peculiarity of this worm. That is, it opens near the mouth of the worm. See here, this is the male. In the male, you will see, say, this nervous system, you can see all this circumference that are present, all the nerves. This is the digestive system that is present. See here, the testis, seminal vesicles, spicules, all that. And in the end, you can see the spicule. It is coming out through a preanal and postanal, this area. Between that, in this posterior end, you will see that the spicule is present and there are some sensory, what you call, sensory units that are present here in this specular ends. Okay, and in the females, you can see here, this is the complete digestive system. This side you can see, and above this you can see that this is the uteri which is paired, and it is going to end in the vulva and the vagina. See, this is the ovaries. Usually this is the anus. Okay. Now this uterus, it has become coiled, and this is full of the eggs. This is the seminal recipient. This is the oviduct. This is how it is going to be there. Females, here also, depending upon the different species, different types of production is there, or different ways in which they can reproduce. That is, they can be viviparous. Viviparous means when they are going to produce the larvae. Oviparous, when the worm is going to lay eggs. And in some cases, ovoviviparous, that is, when the worm lays eggs, immediately they get hatched into a larva. That time it is known as ovoviviparous. Fertilization is internal, and juveniles go through several development stages. Each time the molting or shedding of the cuticle is going to occur. These nematodes, they can be either intestinal or they can be present in the tissues. There are different types of species which exist in the intestine or parasite in the intestine, and some are present in the skin. The nematodes are quite diverse, or they have a number of species which are existing and all have different way of functioning, and they have about 1500 species of nematodes that are present in the world. Nematodes can be parasites or they can be even free living. Free living nematodes, they are present in the sea, they are present in the fresh water, as well as they are present in the soil. These nematodes, they occur worldwide in the environments, and most live in the intestinal spaces of sediments and soils. Many root-feeding nematodes are also present, which are the agricultural pests. This species pierce the root cells and suck the contents of the plant. That is how they are going to destroy the plants. Nematodes are estimated to destroy 12% of the cash crops annually. That's how they are hazardous to human beings. Parasitic nematodes. There are great number of species of parasitic nematodes, and they attack virtually all group of animals as well as plants. Parasitic forms include different types of worms like Ascaris, hookworm, guinea worm, trichinia worm, pinworm, and filarial worms. So, we are going to start studying this parasitic forms. Now, in this parasitic form, the first nematode that we are going to study is Ascaris. Ascaris lumbricoides. The common name of this worm, most of you might have heard, that is roundworm. Roundworm infection is very, very common among the children. Not only children are infected, even the adults are equally infected with this Ascaris infection. The Ascaris lumbricoides, they belong to kingdom Animalia, subkingdom is Metazoa, the phylum is Nemathelminths. The class to which this Ascaris belongs is Nematoda. Ascaris lumbricoid is the species name, and Ascaris lumbricoid is an intestinal parasite. Common name is roundworm. They are the largest roundworms that are parasitic in human beings. Around 1.4 billion people worldwide are infected with Ascaris lumbricoids, and this Ascaris live in the small intestine of human beings. Ascaris lumbricoids is prevalent in the inadequate sanitation areas and where untreated human feces are used as manure. This is also known as the night soil. So in many places you might have seen that people go in the farms, they excrete there, and that sometimes even the earlier times this feces, human feces, were used as manure. Therefore, this type of infection was very largely common in worldwide. The one of the important character of this small roundworm is that female worm produces around 2 lakh eggs per day. This is the distinguishing character of this particular roundworm. So you can understand that how this infection can spread, because 2 lakh eggs per day is not a small matter. So spreading of infection becomes very, very vulnerable. Okay, that's how many people that are infected. Now, why this infection is so much? The reason is, in many areas you might have seen or you might have observed that people do not have proper hygienic habits. Hygienic habits, what I mean here is, people are not used to wash their hands before the food, before consuming anything. Whenever they are eating something, people have to wash the hands. Just washing hands with water is not sufficient. It should be washed with some soap. Then only this infection can be prevented. Secondly, washing hands after going to the toilet, that is also very important, because if any person is having infection, he will reinfect himself if he is not following the proper hygienic habits. The reason is that infection, and I am telling you, is going to be with all this way. The reason is, infection occurs only and only when the eggs are going into the human being. In going into the human being means what? When the eggs are swallowed by the human being, and this occurs when the food is uncooked, or the soiled fingers are put into the mouth, or you're not washing the hands properly, and your hands may be contaminated with this eggs. While seeing the life cycle, etc., you'll understand what I'm telling you about this infection. Okay? Now let us study the characters of these organisms. As I already told you that nematodes exist. They are dioecious, we call that. Dioecious means male worm is different and a female worm is different. Most of the characters may be same, but yet distinguishing characters are present in this worms. First and foremost, difference is in the length of the male and female worm. That is, the males are smaller than the females. The male is 15 to 30 centimeters long and 2 to 4 millimeters wide. And the female is 20 to 40 centimeter long and 3 to 6 millimeters wide. So, these are more male worms are more thin and thread-like than the female worms. Male are shorter than the female worms. Both are cylindrical in shape. Both are pinkish when freshly passed. Both have got the mouth on the anterior end, and this mouth is surrounded by 3 finely toothed lips. The posterior end of the male and the female are different. The posterior end of the male is curved and it has got the copulatory spicules. Whereas the female posterior end is straight and conical. The vulva lies at the junction of anterior and middle thirds of the body, and the worms are oviparous. Oviparous means the legs, and earlier when I have shown you the figures of male and female worms, I have shown you that the males and the females can be differentiated depending upon the posterior end of the worm. 2 lakh eggs per day are laid by the female worm, and this eggs can be either fertilized or unfertilized. See here, the male worm, you can see that the anus or spicule is present, whereas in female you will see that this is the anus, this is the papillae, and it is straight conical. Okay, the head, the anterior head, it has got 3 lips. I told you. This is the papilla, that is sensory area. This is the papilla, which is the sensory area. These are the 2 ventral lips, and this is the dorsal lip. Secondly, the female which lays 2 lakh eggs per day, they can be decorticated eggs, they can be fertilized eggs, or they can be unfertilized eggs. Now, this decorticated eggs are the eggs, they are not going to get, or embryonated eggs. They are not useful, they are of no use for continuing the further life cycle. How can you make out this different types of eggs? That is, the decorticated eggs, they do not have the albuminous coat around them, and the fertilized eggs, they have got albuminous coat around it. Shell is present in all the 3, but the shell is more thick in the fertilized eggs. The unsegmented ovum is present in both decorticated and fertilized eggs, and you can see the clear crescents are present in the both the poles of the cell. This is the crescent, the clear area that is present there. The unfertilized eggs, they may or may not have the albinous, the eggshell thicker one. It is quite thinner one. And the albuminous coat is also very irregular. Now this is one, another. See, this is how when they are soiled you can see. Okay, fertilized, decorticated egg, how they look, fertilized egg, how they look, and unfertilized egg, how they look. The unfertilized egg, it has got a trophic ovum in it. Whereas this decorticated and fertilized egg, they have the ovum that is nicely found in it. Okay, or developed type of form, you can say. Now, these eggs of roundworms, I had told you that they are fertilized and unfertilized. Now, how you can differentiate? That is depending upon the size of the egg. The fertilized eggs are smaller than the unfertilized eggs. 60 micrometer by 40 micrometers is the size of the egg. They are yellowish brown in color because they are bile-stained. The unsegmented Ovum is seen in the shell. The shell is quite thick. There's a clear crescentic space between the ovum and the eggshell at both the poles. This eggshell, it is covered by the uneven albuminous coat if cortical, and no albuminous coat is there, then you will see that it is absent. Means if albuminous coat is not present, this egg is known as the corticated egg. Egg floats on saturated solution of common salt, and once this egg pass into the soil, the development into the larvae takes around 30 to 40 days. And after the development is completed in the soil, then only this fertilized egg is infective to the humans. This fertilized eggs, they are resistant to drying, but they are killed by the direct sunlight. The unfertilized eggs, they are 90 mm by 45 mm. They are bile stream. The ovum is atrophied in the eggshell. It is disorganized and having highly refractile granules which are of various sizes. There is no space between the eggshell at the poles. So all this ovum or atrophied ovum that is spread throughout the shell, within the shell, they are disorganized, they are granular. Albuminous coat, if present, it is very, very thin. Even the eggshell is very, very thin. And this unfertilized eggs, they are not going to float on the saturated solution of common salt. This is how you are able to distinguish between the fertilized and unfertilized egg. Two things are there on which you can make out whether it is fertilized egg or an unfertilized egg. Fertilized eggs, they are lighter, smaller, and therefore they will be floating on the saturated solution of common salt. This unfertilized eggs, they are larger, they are heavy, and the size is also big. They do not float on the saturated solution of salt. Now coming to the life cycle of Ascaris lumbricoides. Earlier in this, protozoans you have seen one or two hosts, intermediate hosts, etc. are required to complete the life cycle. Only then the life cycle will be complete. They have to reach a particular host, they have to reach the intermediate host, and that's how it has to be completed. But in phylum nematodes, usually only one host is required. The life cycle is also very simple. The infective forms are larvae. Larva means embryonated eggs. The embryo is present within the egg. That means whenever the egg breaks, the adult forms will come out. Now, infection occurs in the human beings only when the embryonated eggs are ingested. And the injection of these eggs is through contaminated food, drink, raw vegetables, or from fecally contaminated hands. So, in the introduction, I had told you, if you remember, that the hygiene is an important thing that is required to be away from infections. If a person does not have the method, the hygiene method that is to be followed, that is washing hands with soap before eating. Secondly, washing hands with soap after going to the toilet. Thirdly, washing all the fruits and vegetables that are bought from the market 2 or 3 times with water. Then only the eggs can be removed, or contamination is, you can say you are away from contamination because you remove it by washing. Many times it is advisable to use salt for washing this fruits and vegetables, because if you are putting little bit of salt, whatever the eggs are, they will float and they will move out of the fruits and vegetables. Sanitation is also very important. Now, in sanitation means the toilets, etc., should be kept neat and clean so that the fecal matter, if an infected person passes, the embryos or the eggs that are there in the feces of the person will not come out or spread to the other people. Eggs, once the person is contaminated or infected with this embryonated egg through the contaminated food or drink, eggs are swallowed. They reach the stomach where the gastric juice is secreted, but the eggs are not going to break the shell of the eggshell is not broken there, because there is no action of gastric juice over the eggshell of this Ascaris. They are thick ones, so they will not break there. But once they pass the duodenum and reach the intestine, at this area they start thinning. And once they reach the small intestine, the eggshell breaks and the larvae are hatched. This larvae, once hatched, they are motile and they will penetrate to the mucosa and the portal circulation and reach the liver. In the liver, this larvae will stay for 3 to 4 days, and from there they are going to enter the systemic circulation. Once they reach the systemic circulation, it is easy for the worms to reach in the lungs. Once they reach the lungs, there they are going to stay for 10 to 15 days. They will develop there and they will become 0.2 mm to 2 mm after molting twice on the 5th and the 10th day. Here in the lungs, they get converted from the motile larva form to the rhabditiform larva. And this rhabditiform larva, they will penetrate through the capillaries and enter the alveoli. Once this worm is entering the alveoli, they are easily coming into the bronchial tree by ascending the alveoli. After ascending through the bronchial tree, they reach the trachea, larynx, and pharynx. The larva then will crawl over the epiglottis and swallowed with the saliva, and the larvae passes down through the esophagus and stomach and return to the upper part of the intestine. In the intestine, when they reach, it is around 25th to 29th day the infection has already occurred, and here the worm is going to grow into an adult form. That is, the worm which was 0.2 to 2 millimeters, that will grow into the adult form. And we have seen the size of the adult worm. The male is 15 to 30 centimeters and the female is 20 to 40 centimeters. So this is the size they are going to grow after molting takes place. The fourth molting takes place. They become sexually active, and this occurs after 6 to 10 weeks. After mating, the female lays the fertilized eggs, and the eggs are passed in the feces around 60 to 70 days after initial infection. So, it is around more than 2.5 months that the whole cycle completes, and the patient is going to excrete the eggs in the feces. The female worms that have never been fertilized, they are going to give the unfertilized eggs. This is because of exhaustion of the sperm that has been produced, and therefore the fertilization of the eggs does not take place. When the eggs reach the soil, if they are embryonated, then they reinfect the human being and the cycle continues. One important thing which you have to remember is the larva undergoes molting 4 times to complete its life cycle. The first molting will take place outside the human being, that is, when in the eggshell itself, before it is being swallowed by the human. When it is in the soil, that time it is going to cause the first molting. Second molting will take place in the lungs. Sorry. The second molting will take place in the lungs on the 5th day of infection, after reaching the lungs, and 10th day. And last, that is the fourth molting, will take place on 25th to 30th day of infection in the small intestine. Is the whole thing here you can see how in the feces fertilized, unfertilized eggs are there. They are passed, they are getting embryonated on the 3rd stage, and when this embryonated egg is taken by the human being, they will reach the stomach, and the larvae are released in the intestine. Then they are coming into the portal circulation, systemic circulation, reaching the lungs. From lungs, they are coming to the trachea, epiglottis, coming back to esophagus, stomach, and intestine, and getting after molten fourth time in the intestine, the adult worms are released, and when this adult worms they are mating, the female is going to lay the eggs, which are around 2 lakhs per day, and when these eggs are laid, they are passing through the feces outside into the grass or wherever the excreta is put. From there, the eggs are going to get spread, stay in the soil for days together. They will not be destroyed by the other environmental factors except the direct sunlight. Okay, and therefore they are able to infect the human beings when ingested. So this is how the child or the patient is going to look. You can see the stomach, how it is bloated, and this is how, if the infection is heavy, they are excreted. Pathogenicity and clinical features of this Ascaris. Ascaris lumbricoides, they are going to cause ascariasis. Clinical manifestation of ascariasis are due to both larvae and adults. So, larvae will produce different types of infections or clinical features, and the adult worms will produce different type of clinical features. Now, you know that the larvae, they are migrating in the body, in the portal circulation, in the systemic circulation, etc., in the stomach, intestine, and all. So this migrating larvae, they are going to cause certain clinical features. What do you mean by clinical features? The signs and symptoms that are produced. Okay. So this migrating larvae in the lungs, they will produce symptoms like non-productive cough. Cough is there. There is no cough. There is no you call sputum, that is seen. They will also cause the substernal discomfort and dyspnea, that is difficulty in breathing, because once they are present in the lungs, we have seen that they are going to develop from 0.2 mm to 2 mm there, and they are staying in the lungs for 10 to 15 days. So there they are going to take molting. They are going to have molting for 2 times and get converted into an radiform larva. So the manifestations are quite common when the larvae are present in the lungs. When those larvae are going to penetrate the lungs, the alveoli are penetrated, then the blood is going to ooze out. And when the person or when the patient is going to spit, the sputum can be blood tinted. So the expectoration of that patient is going to be blood tinted. That is the sign we come to know that some infection is there in the lung. The person infected with larva can show fever, exhibit fever. He has got eosinophilia and urticaria. Urticaria means scratching that is seen all over the body. And all this is because the larvae are penetrating the alveoli. The larvae have got antigenic material which is there, which is going to cause irritation in the blood, and you will see that this penetrating larvae will produce hemorrhages and induce eosinophilic response in the host. Therefore, the eosinophilic count can go above 20 per cubic millimeter in the blood. The resulting eosinophilic pneumonia is referred to as Loeffler's pneumonia or syndrome. So, in pneumonia, there are certain changes that are taking place in the lungs. The lungs become heavy, then the alveoli are broken, then blood is oozing out, liquid is formed, so the lungs become heavy, inflamed, and because of that, the alveoli penetration and all, and the oozing out of lung, the person is feeling discomfort at the substernal region because of the heaviness of the lungs, and the person is having difficulty in breathing because the air spaces are blocked. So these are the features of pneumonia. There's a fever, there's pain in the chest, discomfort at the substernal region, dyspnea. Because of this infection or eosinophilic pneumonia is referred to as Loeffler's pneumonia or Loeffler's syndrome. When the larvae passes beyond the pulmonary circulation, they will reach the systemic circulation. So once they are reaching the systemic circulation, the blood is going to go throughout the body and into various organs. When this larvae reach, they can show different types of clinical manifestations. These clinical manifestations are because of the occlusion of the small blood vessels that are seen. And this occlusion of the small blood vessels in the heart, brain, and kidney are going to produce the clinical manifestations, and this can be seen on the X-rays. The infection of the adult worm may cause a symptomatic infection. When the large number are present, especially in the children, they will interfere with the proper digestion and absorption of the food. This results in protein, energy, malnutrition, and vitamin A deficiency. As you all know, that absorption of the nutrients is taking place in the small intestine from the epithelium. And when when the roundworms are present in the intestine and they are damaging this epithelial tissue, the absorption has been hampered. And therefore, the nutrients are not absorbed, vitamins are not absorbed, and therefore the patient is developing protein, energy, malnutrition, and vitamin deficiency. So these worms take more of vitamin A, and therefore you will see that patient is suffering from vitamin A deficiency. When large number of entangled worms are there in the intestine, they can lead to obstruction of the intestine. So the lumen has been obstructed, and therefore the number of worms that are there in the intestine, the quantity and the length, what we have seen that they are 30 to 40 centimeters long. And just imagine that we have also seen that number of eggs that are being laid by that. So intestinal obstruction occurs, and this is going to complicate. Now these worms, they are usually going to attach to the intestine, and their movements is causing, when so many worms are there and they are moving, they will cause the flow or movement to the intestine, and this faster what you call peristaltic movement that is there in the intestine, that is leading to intussusception. Now intussusception is nothing but the intestine, it is getting what you call folded in itself, and volvulus is that wherever the intestine, in the intestine at a given particular place, so many organisms have been infected, they will cause heavy weight and it will start pulling down, and a knot is formed in the intestine, and that is known as the volvulus. Now, intussusception, intestinal obstruction, and volvulus, they are the emergencies caused in the human being, and that is going to cause, or that is going to need a surgery. Surgical intervention is the only thing that can be done in these cases. Single worm sometimes enter into the appendix and cause appendicitis, that is inflammation of the appendix can occur, and many times that also causes a surgical intervention. It may enter this roundworm may enter the ampulla of water leading to the biliary colic, cholelithiasis, cholangitis. Then it can even cause pancreatitis, and rarely it can cause hepatic abscess. So when these worms are going into the common bile duct, then in the bile they can cause the inflammation. That is the bile, what you call the gallbladder, which secretes the bile. Okay, so this gallbladder has been infected, and this gallbladder, when it has been obstructed, leads to cholelithiasis, cholecystitis, cholangitis, and all this because of the worms that are going to obstruct there. Then, when the worms go in the circulation and they are blocking the blood vessel of the pancreas, then pancreatitis is caused. That is inflammation of the pancreas. Sometimes you will see intrahepatic abscess. Now, intrahepatic abscess means what? That when the worms are present in the liver, they have got the habit of penetrating. So, the architecture of this liver, it has been broken down, and the abscess is formed in that particular liver, and this is known as the intrahepatic abscess. When the worms are going to penetrate the intestine, because of thinning that has occurred due to the presence of the ulcers, sometimes ulcers are just like that only, that is, the person is having that tendency of formation of the ulcers. Sometimes the person might have suffered with typhoid and typhoid ulcers are present. Sometimes the person has is a patient of tuberculosis. So tubercular ulcers are present in the intestine. So thinning of the intestine has occurred, and as you know that the worms have got the tendency or the quality of penetrating the tissues, it will start penetrating, so they will come out of the intestine, and therefore because of it peritonitis occurs. This adult worm may migrate through esophagus and may reach the larynx to cause asphyxia. So, I had told you earlier that once they reach from the lungs, they are reaching the systemic circulation. Then they are coming via the trachea. They come out. They are swallowed and reach the esophagus. From there they are reaching from the epiglottis. They are going to reach to the larynx. And once they are blocking the throat, the asphyxic condition occurs. And sometimes if the worms are more, they will come out of the esophagus. They are coming out of the mouth. They can come out of the nose, and that can occur. That is possible in heavy infections. Hypersensitivity to the worms. Hypersensitivity to the antigens is there that can occur. That is because the worms are going to have a particular liquid, and this liquid, when it is absorbed by the blood, causes hypersensitivity reaction. That is antigen antibody reaction is seen, and therefore this occurs. The person or the patient can also suffer from fever, urticaria, angioneurotic edema, wheezing, conjunctivitis can be seen in such patients. A fever, as I already told you, whenever the worm's fluid has been absorbed in the blood, antigen antibody reaction occurs, and because of that, fever is induced. Secondly, this antigenic fluid also causes antigen antibody reaction, and its action can be seen on the skin, and therefore urticaria are produced. Angioneurotic edema. Now, angio means blood vessels, neurotic means nerve cells. When the worms are reaching the blood vessels, they are coming out of the blood vessels and they are invading the nerves. Then angioneurotic edemas are seen. Wheezing is seen when the person's lungs are infected. That time, I had told you that dyspnea occurs, difficulty in respiration occurs. This leads to wheezing. Conjunctivitis can be seen in patients, that is, whenever through circulation, even the eyes are infected, that time such things occur. Nausea and vomiting may result from presence of adult worms in the intestine. Now, why nausea? Why vomiting? Again, same thing. Antigen antibody reaction occurs, and it is going to trigger the system, and because of this toxic effect, nausea and vomiting is induced. Body fluid of Ascaris, when absorbed in the blood, causes toxic effect, which gives rise to fever. And fever here is mostly typhoid-like fever, that is step ladder pattern, that is there. Worm masses, especially in children, causes obstruction and perforation of the intestine, and sometimes obstruction of the bile and pancreatic duct, very common in children. Because of that, you will see the bloated stomach, intestinal problems, malnutrition. Children may have vomitings, children may have diarrhea, sometimes they may be having constipation, and all such problems are going to be there. Sometimes the patient's mother will be complaining that the child is eating a lot, but the child is very lean and thin. All such things are seen. Now coming to the lab diagnosis, how can we find out the infection? After seeing the clinical features, after seeing the pathogenicity, you are going to advise the patient for blood examination, stool examination, depending upon the conditions, sputum examination, and sometimes, if required, serological examination, and lastly, skin test. Now, demonstration of the eggs in the fecal sample by direct wet mount or concentration techniques, that is it. So, the fecal matter samples have to be collected. They have to be diluted, and a wet mount of the, that this eggs, they float in the concentrated solution of salt. So you put in that, if the and examine for the eggs by direct wet mount. Take little loop full of sample on this slide, put a cover slip, and by hanging drop technique or just by direct technique also, you can demonstrate the eggs. In concentration technique also, that is the diluted fecal matter, you are going to centrifuge it, and the centrifuge material is taken to absorb for the eggs in the concentrate. If the patient is complaining that the blood tingles, sputum is coming out, there's a cough, and sometimes it is clear, then you take the sputum sample and look into for the larva. Sometimes the patient's vomiters and freshly packed stools, if taken, you can see that adult worm is there. So even if you give an purgative and it is possible that you can demonstrate the adult worm. Blood examination, because it is allergic type of reaction, antigen antibodies reaction is there, eosinophilic count is going to be raised, and that can be seen in the blood examination, 20 per cubic mm. That is the count that can be more than 20 cells per cubic mm, that is seen in the blood count. Skin test, usually antigen antibody reaction by giving a small dose of the antigen. You give on the skin, you mark it, and if there is any swelling that is seen, it shows that antigenic reaction is positive and the person is infected with Ascaris. Serological test, they are of use in Ascaris pneumonitis, and the available tests include ELISA, IHA, and microprecipitation of the larvae. Treatment, pyrantel palmoate and mebendazole are the drug of choice, but these are not of use in heavy infection and cannot be given to the pregnant women. So, in heavy infection, mebendazole and albendazole are contraindicated, and this can provoke the ectopic migration of the worms. Like we have seen earlier, that the ectopic migration means they are apart from the intestine, the worms can go out. So they can come in the mouth, they can come in the nose, they can even travel to the ears, and so on can happen. So ectopic migration is possible. Therefore, you don't give albendazole and mebendazole to the patients. Pyrantel palmoate and piperazine citrate are effective in heavy infection and even in the pregnant ladies. Now, how will you control this infection? It is just controlling is just simple, proper sanitation, filtering of drinking water, washing of vegetables and fruits, avoid use of untreated human feces as manure, and you can also prevent by maintaining all the hygienic habits. Whatever your shoes, chappals, everything that has been soiled outside should not be brought into the house. They should be removed out, because these are the most vulnerable source of infection. Avoid eating roadsides, because the dust which is there on the food that is there in this hawkers or whatever they are selling, that can induce the eggs in your body. Unknowingly you're eating that, all this pani puri, pav bhaji, then your what else, so many in Bombay and all, we see that so many fruits, etc. All the hawkers are there. Even the fooding is going outside, isn't it? Then the sandwiches, etc. All are in the crowded and this areas where the soil is going to be there on that, and they are the main source of infection. That is why when you eat outside, you have problems after a few days. So, eating outside food, proper sanitization, filtration of water, washing of vegetables and fruits, washing of hands before eating, washing of hands after going to toilet, soil hands and legs by the soil should be prevented. This is how prevention and control of ascariasis is possible. Thank you for patiently listening to my lecture.